Hi, AP Bio, it's Miss Bear. And today we're going to do a quick lesson on the endomembrane system. Now, if you are in my class, you should have gotten a document to go along with this lesson. It should be a guide to help you go through this lesson. But then if you're not in my class, it should also be linked in the description box. Couple of things I want to note as we're going through this. I recommend having an elbow partner or a buddy so that you can talk about the questions with you. And if not, make sure that you're pausing the video to take enough time to answer the questions for yourself. If you are answering a question, I encourage you to write it down because it does help solidify the information in your brain. However, it's not a requirement. So, using this video, how would you describe what white blood cells do to get rid of bacteria? We're going to play this video and I want you to see what you're seeing. You basically just have to describe what you're seeing. So the white blood cell is the big blobby thing that's chasing the black dot. All the other little circles are red blood cells. This is somebody's blood sample, and they're watching a white blood cell kind of like heat seek onto a bacteria. Now watch now what happens. So how would you describe, take a second to describe what the white blood cell did to the bacteria to get rid of it? Take a second, write it down. Hopefully you had a chance to write it down. If not, make sure to pause the video. And we're going to talk about it. What this white blood cell did was a form of endocytosis. And cytosis is a part of membrane transport. So we've been talking thus far about how molecules get past the cell membrane in different ways. And in getting past the cell membrane, there's a lot of energy requirements that play into it. Now, endocytosis or cytosis in general is one of those ways that things can get into the cell membrane. So if you've been in my class, we've talked about passive versus active transport. We are going to talk about the final kind of capstone, not capstone, but the end of our active transport segment, which is cytosis, which is movement of the cell membrane using vesicles. So let's take a look at this particular type of endocytosis. So watch this particle. It's too big to pass through the cell membrane, but it also needs to get into the cell membrane. The cell needs whatever this molecule is. So what it's going to do is it's basically going to wrap its membrane around the particle and then pinch off the membrane to form a vesicle. And a vesicle is just like a membrane package. Imagine it like a membrane balloon, right? And so this is another image of a white blood cell doing endocytosis, where it's swallowing up a bacteria. Okay. It's the same thing we just saw. Another one. Here is a amoeba, which is a large single-celled organism, and it's endocytosing a paramecium, or excuse me, what was that? That was a paramecium. Yeah, it's a paramecium. So these are just a couple of the ways that the cell membrane can be used to not just transport things across the cell membrane, but also help an organism get around. Okay. So let's use a scenario. Let's say that we are like going to go see our significant other after school and you give them a hug. Now, the feeling of warmth and safety that you experience with that person is because of the molecule that you see on the screen. It's because of this molecule called oxytocin. Now, the reason that hearts are in this molecule is not that there's actual hearts in the molecule. It's because this molecule is often considered the love hormone. This molecule is associated with increased bonding. Associated with reduced aggression in folks, reduced stress and anxiety, and increased relaxation and well-being. It's also a critical component of childbirth, which I encourage you to research more if you're interested. So looking at this molecule, this is a quick macromolecules review. So knowing what you know about macromolecules, carbohydrates, lipids, nucleic acids, amino acids. Which type of macromolecule do you think oxytocin is? take a guess. Oxytocin is a peptide. It's a protein. And you can tell because it follows the carbon-carbon-nitrogen backbone. You have the carbon-carbon-nitrogen, carbon-carbon-nitrogen. It's just wrapped itself around itself. And you can see with the disulfide bridge there, it's wrapped its linear structure to make it a three-dimensional shape. Now, this is what's called a neuropeptide, which is a protein produced in the brain to send signals to the brain and the rest of the body. So this is a little bit of organelle review. If oxytocin is a protein, what organelle do we need to use to start making this molecule? Hopefully you've had a chance to review your organelles if you're watching this video. Now, in order to answer this question, I'm going to go about a long-winded answer because I want you to know exactly how oxytocin is synthesized and then how it will be transported to go throughout the rest of your body and cause you to feel all of these things. So to get us started, we are going to look at the endomembrane system. Now, if you are in my class, you should have a copy of this, and I encourage you to draw on this copy for your notes so that you can keep track of what's going on, okay? So the endomembrane system consists of a variety of different components. It consists of the nucleus, the endoplasmic reticulum, which is up at the top here. It'll be labeled in just a second. The Golgi apparatus, vesicles, lysosomes, and then the cell membrane. Those are all some critical components of the endomembrane system. Now, why is it called the endomembrane system? Well, it's because all of these structures are made of the same membrane. All of these structures are made of the same membrane, and we're going to look at an example of that in just a minute. The first step in this process is going to be to convert DNA into RNA for oxytocin. So this DNA strand here represents the DNA strand for oxytocin. In order for us to get the protein for oxytocin, we're going to make the RNA strand that correlates to that DNA strand. Now, there's a whole separate unit on how that process works. You don't need to know about it right now, but you just need to know that it happens. The next step is going to be for the RNA that was just synthesized in the nucleus to leave the nucleus via these little pores in the membrane, and then it's going to attach itself to free ribosomes. Now, free ribosomes are compared to bound ribosomes, which we're going to talk about in a second. But basically, free ribosomes are floating around in the cytoplasm. These free ribosomes will then translate that RNA into a protein. Now, what are those proteins going to do? Those proteins are going to go into the cytoplasm and perform cell functions. They are going to produce membrane transport molecules. They're going to move things around the cell. They're going to attach themselves to other organelles in order to help with the function of those other organelles. So there's a lot of different functions that these enzymes, or excuse me, these proteins that could be enzymes, that they will do. Now, the fourth and fifth step are very related. They're very close together here. So the fourth step is going to be where the RNA is actually going to leave the nucleus and go to a different location. The RNA is going to go into the endoplasmic reticulum. The endoplasmic reticulum is where ribosomes that are bound ribosomes are going to be stuck to the membrane. And these bound ribosomes are going to take the sequence of RNA and turn it into protein, but it's going to be different proteins. And these proteins can do one of two things. They can either embed themselves into the membrane of the endoplasmic reticulum, which is what you see in number 6, or they can be found in the actual matrix of the endoplasmic reticulum, which is like the cytoplasm of the endoplasmic reticulum. It just means the inside of the endoplasmic reticulum. Now, these components of the endoplasmic reticulum, where there is a protein attached to the membrane, those will become a vesicle. And basically, it will go through the reverse of endocytosis. It's called exocytosis, and it will create a vesicle that is a membrane-bound package of proteins. Now, if they go from the endoplasmic reticulum, they're also going to hit the Golgi apparatus. And the Golgi apparatus is where they are going to be converted into quaternary structures. This is where the proteins will get stuck together and get turned into quaternary structures. Okay. They will get different subunits and then put those subunits together. Now, once the Golgi apparatus has essentially packaged the protein appropriately, it's going to form another vesicle on the other side of the Golgi apparatus. And then that vesicle will pinch off, again through exocytosis, which I'm going to show you in a second, and form a new vesicle. So this is the process of exocytosis. Oops, sorry, the music is really loud, so we're not going to play the music. So the vesicle is here going to have stuff that it's trying to get out of the cell. Now, instead of trying to push it through the cell membrane, it's going to use a vesicle, like a protein. Okay, so a protein is too big to diffuse through the cell membrane. And so the membrane of the vesicle is going to fuse with the membrane of the cell, and it's going to push out whatever it is that it just made. And so that's going to be the protein that we just synthesized. Okay, so here's a couple of examples of that in real life. You can see exocytosis happening down at the bottom. It's a little hard to see in this image, but you can see that it is releasing things. And I encourage you to go back to this video if you're interested in how that process works. So that's step 9, where the vesicle is formed. Step 10 is going to be where the vesicle merges with the cell membrane. And I've divided step 10 into 2 parts, A and B. Step 10 is going to be where, 1, the membrane proteins, the proteins that are embedded in the cell membrane, stay in the cell membrane, and then they can complete their function. They can, allow certain things into the cell, allow certain things to get out of the cell. They are going to have a variety of different functions. But if the proteins are found inside the vesicle, they can then diffuse out of the cell membrane and then get inserted into the blood vessel. Now, I want to remind you what the beginning of this conversation was about. It was about the molecule oxytocin. Well, oxytocin is a protein, and that oxytocin molecule is going to end up moving through the endomembrane system to the blood vessels, where it's going to go through the rest of your body. It's going to go to your heart, where it slows down your heart rate. It's going to go into other places in your brain, where it changes the brain chemistry. It's going to go into a variety of different locations. And in those locations, it's going to cause the physiological effects that we saw originally: the reduction in stress, the increase in relaxation. So this is what I wanted to show you, though, is that this is the membrane that all of these organelles are made of within the endomembrane system, okay? So what this is showing is it's showing the inner membrane, or the inner phospholipid layer, and the outer phospholipid layer. So these proteins, this is what's called a glycoprotein, which if you know anything about root words, you can probably predict what that is. It's a glyco, or a sugar attached to a protein. And that glycoprotein is embedded in the membrane. Now, there's other proteins that are just found in the vesicles. But once they leave the endoplasmic reticulum, they can then merge with the Golgi apparatus, and then they can become modified. Now, this is still the Golgi apparatus up here. And once those proteins merge with the Golgi apparatus, those vesicles can form again after the protein has been modified, and then merge with the cell membrane. So this is just a different way of looking at the diagram that we just drew. So I want to show you what this process looks like in a little bit of a better animation. I wanted to go through it slowly so that you had time to absorb it, but this is what it looks like if you were to actually observe it.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ.
So I'm going to skip ahead a little bit. I wanted to get you that review of your prokaryotic versus eukaryotic cells, but I want to skip ahead to the point where it's talking about the endomembrane system.
Nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials.
So that's going to be the end of the endoplasmic reticulum, or excuse me, the endomembrane system section, I believe. There are other organelles that this video talks about that's really interesting and has really good animations, but I want to focus on the endomembrane system for today. So that being said, we are at the end of our lesson, and at the end of the lesson, there are a series of review questions. Some of those questions are on the screen right now, but they are also on the handout if you would like to look at the handout. There's a couple of different questions related to the endomembrane system. So with that, I hope that was helpful. And if you have any questions, again, as always, don't hesitate to ask.